If flies, which are wild type for both body color and wing shape, are crossed with flies which are black and vestigial, one will observe a 9:3 to 3:1 ratio in the F2 generation if these two genes assort themselves independently. This would occur if the genes are located on different chromosomes, or if they are located far apart on the same chromosome. Crossing over could still allow for independent assortment. If, in this cross, the genes black and vestigial are completely linked, either because these two traits are caused by one single gene, or the two genes which cause these two traits are very close to each other on the same chromosome, then one could observe a 3:1 ratio in the F2 generation with only two phenotypes: flies which are wild type for both body color and wing shape, as was one of the parents in the parental generation, and then flies which are both black and vestigial, as was the other parent in the parental generation. So the F2 would have only two phenotypes if these genes were completely linked. If, however, the genes were linked, but that crossing over could occasionally separate the alleles, then there would be occasions where black could be present in a gamete but not vestigial, or vestigial could be present in a gamete but not black. And as a result, while a 9:3 to 3:1 ratio would not be observed, one could observe phenotypes other than the two parental phenotypes, in other words, the recombinant phenotypes. The farther apart that these genes were located, the more frequently gametes would have one but not the other allele. In other words, more frequently would crossing over separate these two alleles, this linkage group. And so, the frequency with which crossing over separates a linkage group can be used to map chromosomes, because genes which are more closely linked are less likely to be separated.